everyone, and welcome to today's edition of the SCHOTT Pharmaversity podcast. My name is Neus, and I'm going to be your host. Today, we will talk about large volume subcutaneous injections. These kind of injections are typically defined as doses exceeding 2 milliliters and represent a growing area in the pharmaceutical drug delivery. To learn more about this topic, I have with me Robert Lindner. Robert is Global Product Manager at SCHOTT Pharma, and he has been dealing with ready-to-use containers, both vials and cartridges, and their sterile packaging since several years. Hi, Robert, and welcome back.
Hi, Neos. Thanks. It's always a pleasure to talk with you in the podcast.
Great. So happy to have you here. So I'd like to explore with you a little bit this large volume subcutaneous field from different perspectives and learn more about that. And to start with, why are we talking more and more about these large volume subcutaneous injections currently? So what are the drivers behind this growth?
That's a very good starting question, Neos. So there is really a large shift in the direction of large volume subcutaneous injections. And this is largely driven by advancements in biologics, advancements also in high concentration formulations, and also an increasing need to transition therapies from clinics to a home setting. So this is really not only about reducing the healthcare costs because you need less time from healthcare professionals, but also this increases the patient benefits because the patient can administer critical medication, especially on a regular basis, in his own home, and he does not have to visit hospitals to manage his chronic conditions. And this is really what it's all about, right? Making diseases better to manage, improve patient compliance, make it more convenient, make it more convenient, and also, reduce the dependency on these clinical settings. And you can also, if you allow me to expand a bit on that, you can also see that in the approval numbers, right? So approval numbers for biologics in the large volume space. And there you can really see that in the last years, large volume subcutaneous injections, the approvals for them have significantly increased in the last 5 years, especially for monoclonal antibodies. And their, main targets are cancer and autoimmune diseases. So there's really an evolving trend. And I think this will continue to expand in the coming years.
Yeah, indeed. We also see or can track very good this continuous increase in the approvals. So you've mentioned a couple of minutes ago about that there are technical advancements that has enabled to make possible these large volume injections to reach the patients. Could you provide some examples? What exactly are you talking about?
Yeah, so first in general, there has been a significant process in developing these injection systems to also cope with higher capacity or higher volumes. So they can handle larger volumes, higher viscosities, also ensure more efficient and comfortable delivery. Yeah, so speaking about delivery comfort, that's especially injection site pain. And then, of course, there are also innovations on the horizon, like electromechanical autoinjectors, right, that can adapt to varying viscosities depending on which drug you're putting into it. And then also the advancement with primary containers, such as prefilled syringes and cartridges, they're really the key to these advancements. And maybe just to cite a recent study by SHL, that has really shown that 5 ml injections are feasible with minimal side reactions and swelling. Yeah, but for this, I think we should listen to the experts. So my not direct colleague, but my partner there, Gary Calderwood from SHL, who will detail it more in our Pharmaversity webinar.
Right, I'm really looking for that. So Robert, so if we think about drug delivery, okay, we need the device, but it's not only about the device. What about the primary container, which is within this device, inside the device? How can you support the audience in the selection of that?
So of course, the primary container is highly critical. You can, or I always like to think of the primary container to being kind of like the adapter that translates that adapts the drug formulation and the device so that the drug can be properly injected by the device. And of course, the primary container is also very crucial because it needs to contain the drug and also protect the drug over its lifetime. And at the same time, you don't want to have any interaction of the container with the drug in the ideal world, right? So you want to have a primary packaging container that leaves the drug unharmed, right? So you want to fill the drug, then store it for, I don't know, 2, 3, 4, 5 years. And when you inject it, you want to have the drug in the exact same condition when you at the moment when you actually prepared and filled it into the container, right? So the mechanical performance should be the same, but also the chemical performance should be the same. And for this, there are numerous factors, that are important. So, for example, protein stability is important, viscosity, extractables have to be considered. So the ions that are the ions that are going out of the glass container into the drug over time. And if you look at proteins, so like monoclonal antibodies, they are highly sensitive to changes in the ionic strength. So when over time ions leach out of the primary packaging container, there's a change in the ionic strength and potentially also the pH value. And this, of course, influences the protein stability. And what we've also seen, but I think which is also very logical if you think about it theoretically, is that larger containers have a lower surface to volume ratio, right? So they have less surface to the volume that they can contain, and this by nature reduces the leachable concentration in them, right? Because you have a higher dilution of the potential leachables going out of your package. And this, of course, is beneficial to drug stability. And then, of course, siliconization is also a very interesting topic.
Okay. Can you tell us a little bit more why you're mentioning the interesting topic of siliconization?
Yeah, sure. I just didn't want to make it too much of a monologue, but siliconization, no, really, siliconization is of course a big topic, because as leachables or ionic or ions, silicones also can interact with the proteins, and in the worst case lead to protein aggregation and also particle formation. And what we have, for example, in our ready-to-use cartridges, so our cartridge products, is a baked-on process for the silicone. So we're baking on the silicone, making it a lot more stable. And this then results in very low total free silicone, especially if you compare it to an industry standard glass syringe, right? And this is mainly driven by the fact that the silicon-oxygen covalent bond, which is created during the bake-on process, is around 20 times more stable than the hydrogen bonds, which are typically adhering the silicon emulsion onto the glass surface, right? So when you have a spray-on silicone, then the hydrogen bonds adhere the silicone emulsion onto the glass surface. But when you do a bake-on process, of course, it adheres much better. Yeah, but of course, there also it highly depends on the process. I mean, there's also cross-linked silicone and other ways to stabilize it. But in principle, you can derive that from let's say simple high school chemistry. Yeah.
Yeah, this was what we were reminding me of with chemistry lessons in high school. Yes, indeed, an interesting topic and of relevance. So now you've talked a little bit about this chemical side, the potential effects on the chemical side. You mentioned before the mechanical side. What would be potential mechanical factors that we need to consider?
So mechanical factors like container dimensions, tolerances, break, loose, and gliding force, they are also highly crucial for successful delivery via an injection device. Right, so you need to ensure that there is a mechanical fit. You need to ensure that the break, loose, and gliding force is as expected, because especially when you're using a spring-driven injection device, then the spring is adjusted to a specific gliding force, right? So you need to account for that. And that's why we are closely collaborating with the leading device manufacturers in order to ensure this mechanical compatibility right from the start, right? So it doesn't make any sense if we say, hey, we have a greatly standardized cartridge and it adheres to all the relevant standards if it doesn't fit to the exact device and also the drug later on that is being filled in there. So we need to take close attention to that. And the reasons behind this, as I briefly touched it already, is really that we need to ensure proper dosing. We need to reduce the risk of breakage. So of course you don't want to have too high pressure in the device in the end, right? You want to have a consistent dosing, and especially in devices where the containers can be exchanged, there it's also about user experience. So when you exchange the container, so especially the cartridge within an injection pen, of course, you want the cartridge to always fit nicely into the injection device. So it should not be too hard to put it in, but also you don't want it to have too much wiggle room, right? To reduce error and also patient comfort in the end. Increase patient comfort.
Absolutely. Okay, so you mentioned the cartridges before. For me, if I think about a cartridge, I think cartridge, multi-dose or insulin, 3 ml. So are they typically containers for such large volume injection devices?
So you put it correctly. Cartridges are mainly used for multiple variable doses. The classical example are insulin injections, but there's also rising interest for large volume applications. And this is especially driven by highly sensitive biologics, where, for example, we see an increased risk of needle clogging. And in a cartridge, the cannula is only inserted right before the injection, right? And therefore, the risk of needle clogging is minimized, minimized as much as possible if you really go into the details. And then again, it highly depends on the drug formulation, which device and container combination is chosen. It also depends on the patient needs. So I wouldn't say the cartridge is the best option. It's a very good option, obviously, but also other container formats, like polymer or glass syringes that SCHOTT offers are viable options to deliver large volume drugs. Yeah, so it's really the whole injection device system that consists of multiple components that really drives the decision.
If I think about your role, SCHOTT Pharma, so global product manager for cartridges. So what would be then your vision with, let's say, the cartridges for this large volume field, which is growing and growing?
Oh boy, that's a very big question.
This is a wish list.
Yes. No, of course. What's my vision? No, really. I mean, if 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 you ask me why are we doing that, right? I mean, it really boils down to enable that that patients are enabled to safely and efficiently administer medications, ideally in a home setting. Yeah, as I said before, because it's just much more comfortable for them to do it at home in their used environment. And then also, of course, the developments on our end are really tailored to enable more options to deliver this critical medication. So that's also why we designed the whole cartridge platform to enable injections currently from 1.5 to 10 ml. And we've all based it on existing ISO standards to make it as compatible as possible with other RTU products. Of course, predominantly from SCHOTT IQ, yeah, but really base it on proven industry standards so that it's very easy to also to integrate these products into the different production processes and just get the medication quicker to the patient.
So thanks, Robert. Thank you very much for this interesting talk we had and for guiding us through these market drivers, latest advancements, and so on within the field of large volume subcutaneous injections. I wish you success with the developments and hope to have you here again soon. Thanks, the
Anytime,
audience.
Niels.
Sure.
So
I'm sure we'll have the chance next year to talk again. I want to thank the audience. I started so for listening and for supporting our podcast. We really hope you've enjoyed the podcast and we've triggered some good ideas. We hope to meet you soon again. Bye bye.